Good morning. Today we're going to talk about how organisms use DNA, what it's used for, um, and how that process works. The process being, how does an organism use the information stored in DNA? So last week we learned that DNA is a double-stranded molecule that's made up of different pieces called nucleotides. Those nucleotides have three pieces, which are a phosphate group, a sugar, and a nitrogenous base. And those bases come in four varieties, and those bases carry the code of DNA. The bases come in, for shorthand, A, C, G, and T. Those are the letters that make up the DNA code. Okay, but what does that code tell an organism? Every organism has its own unique set of DNA bases. That's its DNA code, unless, of course, that organism has an identical twin. Maybe some of you have an identical twin. If so, you and your identical twin both have the same exact DNA bases, okay? But otherwise, in nature, every organism that's created through sexual reproduction, which means with two parents, with genetic information from two parents, every one of those organisms has its own unique DNA. So how does an organism use DNA to create its own unique structure? How did my cells use the information in my DNA to create this structure? How did your cells use the DNA in your nucleus to create your structure? How did the oak tree use the DNA in its cells, in its nucleus, in the nucleus of every one of its cells to create that oak tree? Okay, and here's the short answer. Okay, the information in DNA, is used to make proteins. Okay, so my DNA code, my sequence of A's, C's, G's, and T's is used by my cells to make a certain type of protein, to make a certain amount of that protein, and to make those proteins at a certain time. And in doing so, allows my structure to be created. Okay, your DNA tells your cells to make particular proteins in a certain amount and at a certain time, and that creates you. Okay, so how does that process work? Well, let's take a look. Okay, there are basically two steps, and this step right here is called transcription. Okay, and what transcription is, this big pink rectangle is called RNA polymerase. And what that is, that's a protein, and you can see here is the DNA double strand, okay? And that big pink protein, RNA polymerase, basically opens the double helix. You may remember that hydrogen bonds exist between the bases. That was something we explored last week. Those bonds are fairly weak, so they're easily broken apart, okay? So this protein, RNA polymerase it's called, comes along, splits the double helix apart, okay, and reads the bases on one of the strands. It's kind of hard to see here, but this says C, G, T, A, G, C, A, okay? And what it does is it adds a complementary RNA base to the DNA base and then sends what's now a new strand of RNA away from the DNA. Okay, you may remember with DNA, the bases pair in a specific way where if there's an A on one side of the DNA double strand, there's a T on the other side, and those are held together with a hydrogen bond. If there's a C on one side of the DNA double strand, there's a G on the other side, and those are held together with a hydrogen bond. It's very similar when a strand of RNA is created. It's a little different though because there's no T allowed in a strand of RNA. I think that's one of the body's ways of keeping those two pieces of genetic information separate. Also, RNA is different because it doesn't have two strands. It's just a one-stranded molecule, as you can see here, shown in this green strand of RNA. It's only single-stranded. So RNA, there's no T allowed. Instead, it's the letter U is used. Okay, so a DNA C still joins with an RNA G. A DNA G still joins with an RNA C. A DNA T, because T is allowed in DNA, a DNA T joins with an RNA A, but a DNA A joins with an RNA U. Get that off straight? Okay, so if in DNA, let me just say it one more time. If in DNA the base pairs are C and G and A and T, okay, in RNA it's C, G, G, C, T, A, and A, U. So if you look closely at this strand here, you see it goes A, C, U, G, A, G, C, A, U, and you can see here this U is paired with that DNA A, okay, and then this green strand of RNA will leave this situation there, and eventually it will head out of the nucleus into the cytoplasm, okay? That whole process I just described is called transcription, okay? There's two processes involved in turning DNA into proteins. The first process is called transcription, and that is it, where a strand of RNA, it's called messenger RNA, or mRNA, as you can see here, okay, because it carries the message of DNA from the nucleus, from the DNA in the nucleus, to the rest of the cell outside of the nucleus, right? And the rest of the cell exists in the cytoplasm out there, inside of the cell membrane. Okay, so that messenger RNA, or mRNA, is now, after it's been created, being paired up with these DNA bases, it's sent out into the cytoplasm. Okay, once in the cytoplasm, oh, this is just showing that A pairs with U. Okay, once in the cytoplasm, this strand of messenger RNA, which is shown here as this yellow ribbon, okay, is turned into proteins. Now, to understand that, we have to understand that a protein is basically a long chain of amino acids. Okay, you've probably heard of amino acids before. There are 20 naturally existing amino acids, and to be healthy, humans need all 20 of them. Okay, and we can make most of the 20 if we're, or let's say we can make 11 of the 20 if we're given the proper ingredients, but 9 of the 20, okay, we need to make sure we consume so that they're in our body, and those are called essential amino acids. And that's one of the reasons why, if you are a vegetarian, you need to be sure that you are getting the right balance of food so that you get all 20 amino acids in your diet, okay? And there are easy ways to do that, but if you eat meat, you are getting all 20 amino acids. But if you're vegetarian, to get all 20 amino acids, you could have like beans and brown rice. It's classic. You could have beans and toast. It's a British thing. You could have peanut butter on toast. You get all your amino acids. So there are pretty easy ways to do it. Soybeans, which are used to make tofu, have all the amino acids in it. Buckwheat flour has all of the amino acids in it. Quinoa has all the amino acids in it. Anyways, we need to consume amino acids so that we can make proteins, okay? And amino acids here are shown as these green curvy rectangles, and this is hard to see, but it's called glutamic acid, valine, lysine. Those are 3 different types of amino acids. There are 20 amino acids, right? Okay, so let's roll it back. You've got the DNA in the nucleus. The DNA is read by that pink protein called RNA polymerase. the DNA strand is turned into a strip of RNA, a messenger RNA, which is sent from the nucleus. Then that strip ends up in the cytoplasm. Okay. And now the process of translation starts. And during translation, this sort of two-sphere purple snow creature called the ribosome, it's called the ribosome, reads the strip of RNA. Okay, and it reads it 3 bases at a time. So you can see here it's GAG. It's reading that one. Those groups of 3 are called codons. Okay, codons. And it reads 1 codon at a time, or 3 bases at a time. And each 3 base codon tells the ribosome to get a particular amino acid. Okay, so GAG tells the ribosome to get glutamic acid. GUG, we can see, is going to tell, this GUG is going to tell the ribosome to add valine, or maybe already did. Not sure which way this is going. Anyways, you can see then AAG tells the ribosome, AAG tells the ribosome to get lysine and put that into the chain of amino acids. Okay, and that's called translation. Let's look at this next slide, and this will be the last one. Yeah, so it says during translation, amino acids are put into the proper sequence to form the proteins of our body. This is showing the same thing. Okay, in this case, the messenger RNA is this sort of light blue strip, and you can see it's being read by a ribosome, which is itself another protein. And in groups of 3, AAA in this case, the ribosome, that code tells the ribosome to get this particular amino acid, which is called lysine. tRNA, you see here, that stands for transfer RNA. That's another protein that brings the amino acid to the ribosome. We're not going to focus on that much right now. And you can see here's the code GAU, right? We read them. The ribosome reads the RNA in groups of 3. GAU tells the ribosome to get the amino acid. What's that? Aspartic acid. And here comes phenylalanine, and here's tryptophan. Tryptophan you may have heard of. Tryptophan is found in Turkey, among other places, but for a while people thought tryptophan was what made people sleepy after eating a lot of turkey on a Thanksgiving dinner. But now I think scientists think it's probably just because people overeat on Thanksgiving. That's what makes them sleepy. Okay, so that's translation. We're going to leave it there for now. That's enough, and we will pick it up later.